Welcome back to tryptophan catabolism. In the last video, we looked at the pathway of tryptophan conversion to 2-amino-3-carboxymuconate 6-semialdehyde. In this video, we're going to look at the degradation of that molecule, which is shown right here, all the way down to Glutaryl-S-CoA. And then in the video after this one, we'll look at the conversion of Glutaryl-S-CoA down to acetyl-CoA. So ultimately, let's go back and review what we've seen up to this point. Okay, when we used kynureninase in the last pathway, we generated alanine, and we saw that that alanine could be converted into pyruvate, and then that could ultimately be converted into glucose. So what this means is that this particular part of the pathway, at least the part that we've generated alanine in, is glucogenic. The reason it's glucogenic is because we can use the pyruvate to form glucose. Okay, so that's glucogenic. What we're going to find about the last part of this pathway, in which we're going to produce acetyl-CoA, is that it's ketogenic. And the reason it's ketogenic is because we're going to produce acetyl-CoA, and that's going to be used in times of fasting to produce ketone bodies. And the ketone bodies can be used by the muscle, they can be used by the brain, and so forth, much like glucose can. Okay, so this reaction is 2-amino-3-carboxymuconate 6-semialdehyde decarboxylase. And the carboxyl group that's going to get removed is this one that's on the 3 position. So it would be the 3-carboxy group. This is the one that gets removed as carbon dioxide. Okay, and what we generate at this point is something called 2-aminomuconate 6-semialdehyde. Okay, so we generate this molecule, and now we're going to dehydrogenate it using one of the members of the aldehyde dehydrogenase family. And specifically, this is the aldehyde that we're going to dehydrogenate. It's the only aldehyde in the molecule. And so you should expect that if we dehydrogenate it, we should get a carboxylate out of it. And we are, and the carboxylate is present here in 2-aminomuconate. Now, this aldehyde dehydrogenase mechanistically is identical to pretty much every other aldehyde dehydrogenase, right? It has your cysteine thiolate nucleophile in the active site. It has your Bronsted-Lowry base acid proton donor acceptor in the active site. And so it's going to be your typical aldehyde dehydrogenase. And if you need to see the mechanism on that, I recommend that you go watch it. We have that video in this playlist. It might be around here somewhere. But the whole point is we're going to generate an NADH, and then we're also going to generate 2-aminomuconate. Okay? Now, the NADH is not like we're going to get a net NADH because we're actually going to consume it in the next reaction, so it cancels out. Okay, so 2-aminomuconate is going to react with an enzyme not yet characterized. So that's what this ENYC means. It means enzyme not yet characterized. And so if you go to a database and try to look for this enzyme, you won't find it. It has no EC number yet. They haven't isolated it, but we know what the reaction is. It's basically going to consume the NADH that we just produced, basically, and it's going to use water to deaminate 2-aminomuconate. And when it does that, it generates this special molecule right here, which is called alpha-ketoadipate, or sometimes you'll hear it referred to as 2-ketoadipate or 2-oxoadipate. And alpha-ketoadipate is going to get consumed by a member of the alpha-keto acid dehydrogenase complexes. And this particular one is called alpha-ketoadipate dehydrogenase complex. Well, why is this significant? Well, mechanistically, it's identical to the other alpha-keto acid dehydrogenase complexes that we've seen. Well, what are those? Well, that's the pyruvate dehydrogenase complex, the alpha-keto glutarate dehydrogenase complex, and the branched chain alpha-keto acid dehydrogenase complex. So all of those things are mechanistically identical to this one. And if you think back to our study of the pyruvate dehydrogenase complex, you would realize that the first enzyme is our decarboxylase. So this is enzyme 1, and it would be our alpha-ketoadipate dehydrogenase, or alpha-ketoadipate decarboxylase. And then we have enzyme 2, which is going to be dihydrolipoyl transacetylase. And in the process, what we're going to get is this Glutaryl-S-CoA. And in the next video, we'll actually look at Glutaryl-S-CoA catabolism. And then, of course, we have enzyme 3, which is our dehydrogenase, or dihydrolipoyl dehydrogenase. And this is going to use FAD and NAD to regenerate the resting state of the enzyme. And we're going to get NADH out of this. And, of course, the NADH is going to be used by the liver cell that's doing this in order to fuel NADH ubiquinone oxidoreductase, or complex 1 of the mitochondria. So we get an NADH out of this, and that's used to power the pumping of protons by the mitochondrial respiratory chain for the synthesis of ATP. And the Glutaryl-S-CoA that we generate is going to go into Glutaryl-S-CoA catabolism, which we will see in the next video. Okay, so I hope this video gave you a little bit of intuition on how we take the destroyed aromatic ring, which is 2-amino-3-carboxymuconate 6-semialdehyde, and convert it all the way down to alpha-ketoadipate, and then ultimately to Glutaryl-S-CoA. See you in the next video where we actually go into the catabolism of Glutaryl-S-CoA.